Today we're going to be talking about one of the most essential building blocks to life on Earth.
That something being mitosis and meiosis. Okay, let's get into mitosis. Mitosis is a process in which the nucleus is divided and distributed into two new daughter cells. Mitosis can be broken down into four main phases, but before we talk about mitosis, we need to talk about interphase. Interphase is a phase where the cell produces new molecules and organelles. The first phase of mitosis is called prophase. During prophase, the chromatin fibers coil in the nucleus. Sister chromatids join at the waist. Microtubules grow from the centromeres, and the nuclear envelope dissolves. After the nuclear envelope has dissolved, the tubules connect to centromeres and move the chromosome towards the middle of the cell. During the next phase, metaphase, the mitotic spindle has finished forming. The two chromatids pull against the spindle, keeping it centralized. After metaphase, anaphase comes. Anaphase begins with the separation of the sister chromatids. The chromosomes travel to opposite sides of the cell. During the traveling, the cell begins to elongate. Finally, telophase starts when each group of chromosomes are at the opposite sides of the cell from one another. The nuclear envelope reforms and the chromosomes uncoil. After mitosis is ended, there's a period called cytokinesis. During cytokinesis, the cytoplasm duplicates, and a cleavage furrow rips the cell in half, leaving two new identical daughter cells.
Alright boys, next we have meiosis. Meiosis is the process where the nucleus divides and splits into four haploid daughter cells. These daughter cells contain half of the genetic material originally in the cell. These cells in males are the sperm and in females are the eggs. This requires the process to be done twice. They are called meiosis 1 and meiosis 2. In meiosis 1, the homologous chromosomes separate. To kick things off, we have prophase 1. In prophase 1, the chromosomes finish coiling up and special proteins cause the homologous chromosomes to pair up. This leaves us with two sets of four chromatids each. The chromatids in each set exchange corresponding segments in a crossing over. Next we have metaphase 1. A spindle forms between the two central pairs and moves the chromosomes towards the middle of the cell. Now ready to move to opposite sides of the cell, the homologous of each pair break and move apart in anaphase 1. In telophase 1, the chromosomes have now arrived at the poles of the cell. Now each pole should have a haploid chromosome set. Next is meiosis 2. This is where the sister chromatids separate. Meiosis 2 is basically mitosis, except the cells did not undergo chromosome duplication. They align in the middle, split apart, and eventually split into four haploid daughter cells. Sorry, Nick.